As a pediatric oncologist who's been treating patients for over 35 years, it's been really amazing to watch progress. In fact, in 1980, when most of you might not have yet been born, I walked into a research lab for the first time and started what has now become where I am today professionally. And the tools that were available in 1980 to study cells, to study cancer, were absolutely draconian by today's standards, except that they were the beginning. And in pediatric oncology in 1980, people were doing experiments on humans that were changing the landscape for children who had cancer. By that I mean, when a child was diagnosed with cancer in the 60s, you could pretty much say that you could provide comfort care and there was very little hope for long-term survival, except in very few cases. By the 80s, people had started putting together combinations of drugs which were almost unheard of because any of these drugs given by themselves would have been considered to be too toxic to give to a human. In fact, 1980 was roughly, for different cancers, sort of an inflection point where these combination therapies started coming together and they led to these incredible improvements in survival. But the types of drugs that we were giving patients back then were really toxic, and they're still really toxic. We just kept giving more and more of them. For example, nitrogen mustard, which was used in World War I to kill people, literally on the battlefield, was the basis of drugs we were using to treat Hodgkin's disease in 1980. Think about that. We were using a drug that was used to kill people on the battlefield in World War I. We were infusing it into children, okay? So the toxicity of drugs has been now recognized for many years as a huge challenge in childhood cancer. And then there's this gap in the patients, like Dr. Sarangi mentioned this morning, that are not yet surviving, even with all of the poisons and the transplants and all the toxic things that we can throw at them. So it's actually really amazing to me and sad in a way that for so many cancers, we've had basically static levels of survival since the 1980s, 1990s, depending on the cancer. And there's so much more room for improvement. And I challenge folks here who are starting their careers to think about that and to think about creative ways where you can pick off these challenges, the difficult leukemias that Dr. Sarangi mentioned, the brain tumors that many of our speakers mentioned this morning. Those are on the horizon. And for the patients who we are curing, the ones that we're giving still some of these very toxic medicines to, we could go a long way to improve that. It really gives me great pleasure in that context to introduce today's keynote speaker. So I've known Dr. Julia Glade Bender for a number of years, of which I won't say the number. I felt like I wanted to bring her to speak to this group last year, but we had some schedule conflicts, and this year, fortunately, she was able to arrive just from ASCO, the American Society of Clinical Oncology meeting, which finished up a couple days ago. Dr. Glade Bender did her residency at Mount Sinai in New York and then went to Sloan Kettering for her fellowship. And after her fellowship, she had a very long career at Columbia. And for those of you guys who don't know New York City, like we're doing this across the park. And now is back at Sloan Kettering, back across the park. And she has served in so many capacities on committees and in research to try to get new agents into pediatric cancer, that she is literally a treasure trove of information with what has succeeded and what has not succeeded in terms of getting new drugs into patients with childhood cancer. So with that, I want to welcome her and look forward to her talk, telling us how we're going to go forward in the future. Dr. Glade Bender.
So I believe it is still morning, so good morning to everybody, and I am delighted to be here, and thank you to Dr. Turetsky and to CCF for inviting me to be here today. I entitled my talk today, Pediatric Drug Development Getting into Gear, because I was tired of everybody talking about accelerating pediatric drug research, because although we are accelerating, there are also areas where we are, I believe, stalling. And so I wanted to give you a sense of what I see as the trajectory and some things that we need to go forward to do. I am a pediatric clinical trialist, and I try to get new drugs into kids, and therefore I do a lot of consulting work, most of which I won't accept payment for, as well as running clinical trials and serving on data safety monitoring boards. And as Dr. Turetsky was just alluding to, I think pediatric cancer is generally viewed as the poster child for advances in cancer survival. And as Susmita showed us earlier, this is particularly true in leukemia, where you can see that with modern therapy, close to 90% of children are cured. And this incremental progress has really happened because of this iterative process of well-designed clinical trials and clinical and biologic discovery. And we do these large randomized trials, generally speaking, as Dr. Turetsky said, is adding a new drug to the standard of care, which is the best arm of the last study. And then we try to pick off populations. We try to figure out who are the patients who do exceedingly well, and can we give them less therapy with less toxicity? And who are the patients, and what is the biology of the tumor of the patients who are doing less well? And then we repeat the cycle with new drugs with refined risk statuses, and we continue to try to study these outlier populations. This is the process. And I would say it's worked very well for some cancers and not so well for others. And there are still many cancers for which there has been little or no progress, mainly the sarcomas, and I do a lot of sarcomas, brain tumors, and high-risk leukemias that Susmita discussed earlier this morning. And it's really, I focused my career on really this 20% of kids that we are not curing at the moment. And so I decided to make, I don't know if any of you guys used to watch Leno, but my top 10 list of strategies to accelerate pediatric drug development and get our collective feet off the clutch, if you will. And these I will try to weave into a story about drug development as we go along. This is the first time I've ever given this talk, so if I wander a little, I'm very much open to critique at the end of the talk. But these top 10 are legislation. There is a role for the FDA and for actual law that requires drug development in children. Target prioritization, biomarker enrichment, inclusion of kids early on in clinical trials, including those greater than 12 on adult trials, academic and industry collaboration, access to child-friendly formulations, agent prioritization, novel and efficient trial design, combination therapies, and then what is really the topic of this meeting today, pediatric-specific drug development and discovery. So let's start with a history of pediatric drug development legislation. As Dr. Turetsky mentioned, this also sort of overlays my career in pediatric oncology. So I was a fellow from 1995 to 1999, because I did an extra year. So right about that time, in 1997, the FDA actually recognized that there was a problem and that new drugs were not getting to kids. So that was the first legislation. The big change came as I was a young investigator, and this is called the Pediatric Research Act, or PREA, and the Best Pharmaceuticals for Children Act, and that was the first time that actually mandated companies to make a pediatric study plan, or which is actually in Europe now called a PIP, or a Pediatric Investigation Plan. And they mandated it, but they also provided an incentive. They said, if you do the study in kids, we will give you 6 months extra exclusivity rights on your drug. So you can imagine for a blockbuster drug, the first drug that I ever tested, bevacizumab, Avastin, if you have 6 months of patent extension, that's a lot of money. But the study didn't have to be positive in kids, and a waiver could be granted. You could get out of it if it was either an adult orphan disease or if it wasn't relevant for children. So you got your indication in colorectal cancer. And then they sweetened the deal in 2012 when they also passed this rare pediatric disease priority review voucher. And this was a program where they decided that if you could actually get a drug approved in a rare disease in kids, that they would give you a voucher to essentially jump to the head of the line in the FDA review for your new drug application. And if you got this voucher, you could actually sell it on the open market. And selling this on the open market to big pharma has been worth up to $350 million. So this was a very big incentive. Unfortunately, it's actually sunsetting in September. I don't think many people are aware of that. So what did this legislation do, right? So from 1997 to 2017, my colleague Steve Du Bois and his team reviewed 117 non-hormonal drugs were approved by the FDA. Of those, only 6 drugs had initial approvals that included kids, so 5%. And of those 6 drugs, 5 were in leukemia, one of the diseases that overlaps with adults. And 1 was a pediatric drug developed by pediatric oncologists for a pediatric disease, dinutuximab for neuroblastoma. Only a quarter of our studies, pediatric studies, were actually sponsored by industry. And the median time for the first in-human study in adults to getting it into children was 6.5 years. And what if we actually looked at just those last 10 years and actually looked at what happened to those drugs after they were first approved? So of the 78 drugs that were FDA approved in the 10 years from 2007 to 2017, actually all of them were exempt from pediatric development. 52 were adult orphan studies, and 26 had histology-based waivers. So in the end, a total of 8 drugs, 10% of these drugs, ended up over time, cumulative, with a pediatric indication or labeling, and only 20% of them even have any kind of pediatric information on the label. Well, what about the last 6 years, 2018 to 24? Well, guess what? There have been now 30 drugs, cancer drugs approved with pediatric indications. Now you can say, yes, there were also a huge, larger number of drugs, but there were 83 FDA-approved drugs at the time. But this 30 cancer drugs includes some of the drugs that were developed well before that, so I can't give you a percentage. But clearly, there have been 4 times as many drugs approved in the last 6 years than the previous 10. Again, most of these are in leukemia, but that's appropriate for pediatric oncology. That's the most common cancer. And high-risk leukemia is still a big problem. But it's only about 30% as opposed to 80% of those drugs. I have a little coding here on my list that I just wanted to point out to you. So those in bold actually have a full pediatric indication. That means usually somewhere either from 0 and up or 2 years and up. Those that are not bolded, those are only 12 years and up. The one in green, tazemetostat, I counted, but it's 16 years and up. And the ones that get a star, those are the ones that the initial approval included pediatric cancer. So there are 5 of them. So 5 in 6 years versus 5 in, I think it was 17 years. So what happened? So I believe that we do have to give credit to legislation here. So in 2017, Congress passed what's called the Race for Children's Act, or in FDA language, it's the FDA Reauthorization Act, or FDARA. And what did the Race for Kids Act do? It removed that loophole for waivers and orphan drug indications in adults. So now the FDA can mandate that there is a pediatric evaluation of drugs and biologics that have a molecular target that's actually substantially relevant to pediatric cancer. So the indication that you develop it for is no longer the most important thing. It's the target and whether it's relevant to children. And this went into full effect in the year of 2020. There was a meeting of the Pediatric ODAC that I attended, and we actually went through and came up with a list of 200 molecular targets that we thought were relevant to children. And this list exists. It's on the website. I have the link there. And so when you talk to a pharmaceutical company now, you can tell them, you are actually mandated to develop this drug because this target is relevant in children. So this is a big deal. But 200 targets? We don't have that many patients. There are issues here as well. So let's start picking apart the list of drugs that were approved in the last 6 years. And the first point I want to make is that 5 of these were immunotherapy, and specifically immune checkpoint blockade. So starting in about 2014, we noticed the first PD-1 inhibitor, immune checkpoint inhibitor, nivolumab, was approved for the treatment of melanoma. And subsequently, we got very used to seeing these kind of graphs of this incredible long-term durable remissions in diseases that we thought were universally fatal. Metastatic melanoma, lung, head and neck, kidney, breast, bladder, lymphoma. And we were super excited to apply these to kids. And so Crystal Mackel ran a study starting in 2014, ADVL-1412, just to demystify COG names and numbers for people. The first 4 letters tell you what group it came from. The next 2 numbers, the year that the protocol was submitted. The next number is the phase of the study. And the last number is the number of studies, just a counting mechanism. So this was an early phase trial, started in 2015, and it was phase 1. But it enrolled 85 patients. That's a lot of patients. And unfortunately, we saw only 4 responses, and they were in Hodgkin's and non-Hodgkin's, which was an area of overlap with adult malignancy. And no activity in our pediatric solid tumors that we're really looking for therapies for: neuroblastoma, rhabdo, Ewing, osteo, and not even pediatric melanoma, which is a different disease, has different biology. So I use this example to say, actually, some targets are more relevant than others, and children are not little adults, right? And we really need to think about pediatric biology and how relevant is that marker. You can come up with a reason why any biologic marker might have some benefit in kids. But that's really the old way of thinking, I would say, is, oh, the adults have a drug, let's test it. And just to finish up on immunotherapy, we just really have to recognize that childhood cancers are inherently different than our adult cancers. Childhood cancers tend to be mesenchymal, adult cancers epithelial. So we see sarcomas, they see carcinomas. We feel that our tumors are process of a developmental abnormality. They tend to be epigenetic in origin. Theirs are really about aging, about acquiring mutations over time. And therefore, the responder hypothesis for immunotherapy was that there was an increased mutational burden, and those diseases which had increased mutational burden would be seen more as foreign by the immune system and respond to immunotherapy. And the first drug approval in kids, actually, for immunotherapy was for pembrolizumab. It was the first time it was approved with, a drug was approved for the same indication in a disease-agnostic way for both adults and kids for a very rare condition called mismatch repair syndrome. And this is the group of hypermutated glioblastomas that respond to that therapy. A very small fraction, albeit important, of pediatric tumor really only seen in consanguineous, in kids with consanguineous marriage parents in the Middle East. But that nonetheless, a small group. And you compare this mutational burden with the average mutational burden of adult cancers, and you see why these drugs don't really work in kids. But we do have targets that are really relevant in kids, and for over a decade, we've known some of them. So we've known that ALK is an important target in kids for anaplastic large cell lymphoma. In fact, that's where it got its name. Inflammatory myofiblastic tumor, and point mutations in neuroblastoma. We've known that low-grade gliomas are driven by the MAPK pathway, and we'll talk a little bit about that more. And there are other diseases that are also driven by that pathway, like Langerhans cell histiocytosis. And people started recognizing as sequencing was coming along that there were these translocations that happened in kids and caused a variety of diseases. And here we have a kid with NTRK-driven disease, and actually about 85% of infantile fibrosarcomas are driven by NTRK fusions, 100% of congenital mesoblastic nephromas, and then sporadically some brain tumors, some sarcomas, papillary thyroid cancer. And this was the next group of drugs that I really wanted to pay attention to and learn something about how they got developed so we can learn more about efficient drug development. And I put larotrectinib there in bright red because, again, this was the first small molecule, if you will, that was rapidly developed in kids and adults at the same time and approved at the same time in a disease agnostic or a histology agnostic way. It was approved for any NTRK-driven tumor, pediatric or adult. And I just want to sort of, again, put this in context. So the first time I ever saw a dramatic response was this patient pictured here that was enrolled on Yael Massey's first clinical trial of the ALK inhibitor, crizotinib. And this patient had an inflammatory myofibroblastic tumor, and we knew that she had an ALK rearrangement. And the family came to me for a second opinion, and I said, I think you should go on this trial. And they're like, well, we're afraid. We want to go on steroid first. And ultimately, they went to a different, and I sent them down to Yael, and they ultimately went to Boston, who tried steroid. It didn't work, and then she got on the trial. And within 1 month, had this dramatic response. And that trial was set up as really one of the first trials that had a responder hypothesis. We thought that it was likely that patients with either ALK fusion-driven cancers, so anaplastic large cell lymphoma, inflammatory myofibroblastic tumor, the very rare lung cancer that you see in kids, would respond, as well as these point mutations in neuroblastoma that had begun to be characterized. So there was a quick phase 1 all-comer, but if you had any of these diseases that had ALK-driven disease, you could get on this study. And that was a really important change in the way that we were doing clinical trials. And then in the larotrectinib, very similar idea. At first, they did all comers, and they did it at the same time as the adults. There was like no gap, and kids could go on the adult study. And after they saw no responses in patients who did not have this biomarker, they stopped enrolling those patients and really only had to enroll, I think at the time that the FDA approved the drug, 8 patients had enrolled on the study. But you saw these dramatic responses. This is, for those who don't know, a waterfall plot. And anything going down is a response, and anything going up is progressive disease. And this just shows you that all of the patients here on the right side had an NTRK-driven tumor. It didn't matter who the partner was or what the disease was. They all had a response, and none of those without the fusion did. But I want to really look at the comparative timelines of the drugs that were in these categories, okay? So crizotinib, the first one that was developed through the COG, was introduced in the clinic because it was a driver of non-small cell lung cancer in 2017. It was described in 2008 that ALK was actually responsible for familial neuroblastoma. This was a really important discovery, and I gotta give Dr. Massé and all of her collaborators credit, because they quickly developed a trial, the trial I talked about in 2009, and again saw dramatic responses in patients with fusion-driven disease, but not necessarily these point mutations in neuroblastoma. But unfortunately, even though we saw those during the period from 2009 to 2013, the first approval for anaplastic large cell lymphoma did not come until January of 2021. So doing the math here real quick, 15 years later, after it got introduced in the clinic, and 12 years after the pediatric trial opened. Why is that? We'll talk about that a little bit. You compare that to larotrectinib and entrectinib. And here, larotrectinib gets approved in a disease-agnostic way for kids and adults within 4 years. Entrectinib actually gets to the clinic earlier, but it takes them 8. What are the differences here? And I think this is important as we think about how we develop drugs in the future. So the first thing I'd like to talk about is expanding the age of eligibility. And I think this is what was done particularly well with larotrectinib. So the company understood, based on the literature that is out there, there's plenty of it, that a child greater than 12 really doesn't have different metabolic capacity than an adult human. And therefore, the side effect profile is no different, and therefore should be considered an adult in terms of metabolism of drug and safety from the get-go. And there's lots of literature about it, but drug companies still don't buy it. Even the FDA has now put out guidance for industry about including kids older than 12 and up in their trials. And I can tell you that I ask for this every single day, and there's very little uptake. And I beg all of you to pick up this cause. And several people have, including Accelerate, which is an organization, an international organization to accelerate drug development in kids. They call it the FAIR Initiative. And now there's one going on here, and you may hear about it in Washington, run by a patient advocate who is going to call the initiative the Over 12 Initiative. And so look out for it, and I think this is a very important aspect about why larotrectinib was so much faster than any of the other ones. The other point is that you can see that the larotrectinib and the entrectinib, which were shorter, were actually industry-sponsored studies. And there's been some really good literature out there about the importance of an academic and an industry collaboration. And I put the reference here for you about this, which came out in 2022. And basically, it explains, and I think really explained to all of us in the academic community, that academia and industry have fundamentally different expertise and goals. And you really need to work together. It's academia's job to advance the science and to show proof of principle, and then to run the trials that will provide the evidence-based results for a practice change. That's not what industry is interested in. They're interested in discovering drugs and bringing those drugs to market. Now, and I don't mean to say that that's the goal. Everybody does want to cure cancer, but basically that's their role, is they discover the drugs and they get it to market. The thing that the academia really didn't understand is this concept of fit for filing. What does that mean? It means that in order to actually get a drug approved so that we have continued access so that our kids can access drugs, we actually need this package, and we need to collect the data in a certain way, and that costs money. So this collaboration must exist, and they must fund the studies if they want those data. And then the last piece is pediatric formulations. Larotrectinib got out of the gate with a oral suspension for kids, so they could start dosing little kids right away. Crizotinib started out with formulated capsules, and they're trying to target diseases like neuroblastoma, which happen in kids that generally can't swallow capsules. And then they said, well, you could open up those capsules, but it tasted horrible, and the kids just wouldn't take it. And so now there is a pellet formulation, but it's taken years and years and years to develop this pediatric formulation. And so a study I'm working on, I'm just going to pick on selumetinib. This is still a big problem. So selumetinib, and I'll go through it, and I'm going to go over time, I can tell, only comes in 10 and 25 milligram capsules, and we're trying to come up with a study where we can dose escalate and dose de-escalate, and we think we're delivering doses that that we've named, a certain milligrams per kilogram, but in fact we're not actually delivering that dose. We have to figure out our best approximation, and sometimes it means that a BID, a twice-a-day dose, actually one dose is higher than the other dose. Or if we need to go down, we're actually giving a twice-a-day medicine only once a day. And that's very different from a suspension, where you can actually give the dose that you intended to give. It's a very big problem. And I'm going to use that selumetinib example that I was working on yesterday to talk about the next group of drugs, which were the MAP kinase inhibitors. So the mitogen activated protein kinase pathway is very important in pediatrics, and there are a lot of targets. So you can inhibit BRAF with dabrafenib or vemurafenib, and these are called type 1 inhibitors, and they're very important for V600-driven disease. You can inhibit at the level of MEK, and these are selumetinib and trametinib, and this inhibitor has the added potential to block the negative, block the feedback loop that actually happens when you have RAS-driven disease and you only block BRAF. Or you could block down at ERK, or you could combine them. But all of these drugs were originally designed to treat melanoma, MAP-driven adult-type melanoma, except for one, and that would be the pan-RAF inhibitor, tovorafenib, which, based on science done in a lab up in Boston, they showed that this was particularly good for MAP kinase-driven gliomas, not just low grade, but also high grade. And a pediatric focused drug company called Day One bought a shelved asset and developed it for kids specifically, and it was FDA approved 2 weeks ago. So what is MAPK important in? Well, it's important mostly in low-grade glioma. We know that about 2/3 of low-grade glioma is driven by either BRAF V600E or the KIA BRAF fusion. And Brigitte Wiedemann in the audience recognized very well that this might be helpful for NF-driven diseases, but again, started with what isn't really a cancer, but rather a neoplasm, a benign neoplasm that causes significant morbidity, neurofibromas-driven plexiform neurofibromas. And we started seeing with these drugs very early on again, these curves where you see responses in low-grade gliomas. We never saw responses in low-grade gliomas. So this was a huge leap forward. But the incidence of low-grade glioma in the United States is about 3,500 a year. Granted, they live a long time. So while the incidence is low, the cumulative prevalence is actually much higher. And neurofibromatosis is actually the most common genetic disorder, and about 50% of those have plexiform neurofibroma. So these are important drugs. But how many drugs do we need for these indications? Is the first drug to market the best drug? How do we design studies with so few eligible patients? All of the companies now want to develop their drug in these diagnoses. Are there other indications for which we need these drugs? And bottom line is, we're here to treat high-grade cancer. So what do we do when there are so many drugs in the same class? Now we have a different problem. Now we have to prioritize drugs. So this is one of the real functions of this multinational group, Accelerate, that I mentioned. It's an international multi-stakeholder forum that includes academia, industry, patient advocates, and regulators. And they have these strategic forums on single drug targets or pathways or a class. And everybody presents the science, and then drug companies present their assets, and then patient advocates explain the unmet need, and then regulators try to frame the discussion. We have a strategic discussion, and then we write a paper. And in March of 2022, the 9th strategy forum was actually on these MAPK inhibitors. And 10 companies presented 17 assets. And there were 7 PIPs, 3 of which were for melanoma, none of which actually enrolled any patients. And because of this Accelerate conference and the discussion, they actually got out of their FDA requirement to do this in melanoma. There were 12 ongoing studies in low-grade glioma, and this group really tried to redirect pharma to start thinking about other areas of unmet need to develop MAP kinase inhibitors in. So not just low-grade, but high-grade gliomas, Langerhans cell histiocytosis, juvenile myelomonocytic leukemia, and others. And the recommendations were also to focus on the drugs that were substantially different, those that could get into the CNS, those that were less toxic, those that could be given in combination, and really to do this in a more efficient way. So they recommended basket trials that are disease agnostic, platform studies, and really the idea of combinations. So actually, the MAPK pathway is the disease for which I began to understand that not every disease is equally responsive when you have the exact same mutation. This was a New England Journal of Medicine paper put out by Dave Hyman's group just when I was starting at MSK. And they really showed that even though melanoma with V600E was exquisitely sensitive to vemurafenib, if you used this drug in V600E-driven adult cancers, some responded, like non-small cell lung cancer and LCH that we mentioned, but some had no response, showing, again, that this is context-specific. So just because you have the mutation that the drug targets doesn't mean you're going to respond. There's much more biology going on here. And this was called a basket trial. One drug, one mutation, many histologies. And that was the idea behind the NCI match, which was a platform study. A platform study is basically that you have a single design to a phase 2 study. There was a centralized tumor sequencing platform, and if you had the correct mutation, which was a mutation that was picked based on adult data and clinical data showing that that mutation led to response to this drug in some disease, we would have a study for that. And there were multiple pediatric match arms that opened for the different mutations that we knew about and had drugs to target. And this was a very efficient protocol design. We enrolled more patients than we thought we would. And we did enroll to all arms, and you can see that that high blue arm is the MAPK pathway, and there were 3 different drugs at the level of BRAF, MEK, and ERK. So by all circumstances, this was a successful trial. We did what we set out to do. But I show you the updates on the results so far. And that is to say that of the trials that have presented data, there were very few responses. They were mostly in low-grade glioma, and there was 1 high-grade glioma. The ones in white actually enrolled enough patients to complete. The ones in blue had to close early because they didn't enroll enough patients. And so I want to just sort of, if you will, do an autopsy on the study that I ran. Sorry, now I need to go backwards. Which was the olaparib trial. Olaparib is a drug that is used in breast cancer with patients who have BRCA deficiency because it sets up a synthetic lethality because BRCA confers a deficiency in homologous recombination. So what were the clutch stalls in this? Well, first of all, this is exceedingly rare in kids. So only 18 patients out of 1,300 matched to the olaparib arm. And of those, only 6 enrolled on the trial. The biomarkers were borrowed from adults. And I asked at the very beginning, I said, don't you need both genes to be knocked out? Because that is the model in adults. They've lost both expression of both genes in the tumor. And kids, we had seen, only lost 1 gene. And most of the time, it was a germline. So we borrowed these markers from the adults. And because it had never been used in kids before, they made me start at a dose that was 30% lower than the recommended phase 2 dose in adults, even though the dose-limiting toxicity was anemia. Who cares? We'll give blood transfusion. This was very different than what was done in Europe. In Europe, they used a much more multi-omic process. They used a tumor board, and they used this idea of enrichment. We don't know the answer. This is really hypothesis-driven. And so they directed some patients who they thought might respond based on preclinical data, and some patients who just didn't have the biomarker. And they have a trial that actually prioritizes combinations, not single agents. And for DNA damage, they had 2 arms. And the first arm actually enrolled 70 patients. In the same 5 years, I enrolled 6. And they learned a lot from this, and such that when the next trial opened that used olaparib with an ATR inhibitor, which seems to be more relevant in children, they had actually learned enough to know that the DNA repair deficiency genes were probably not the best biomarker for kids. Replication stress was. And so this allowed them, also the trial allowed them to do really careful dose determination, which we weren't allowed to do. And so they actually get results back in an iterative way that lets you begin to understand what the right biomarkers are. Obviously, if you have P53, you don't look like you respond, and there did seem to be some enrichment for DNA damage repair, translocations, and maybe 11q loss in neuroblastoma. So I outlined the key differences, and I just really want to say that the pediatric match, they'll tell you we were going for scientific rigor. Right? We wanted consistent levels of evidence and a clear signal. But the eSMART platform was so much more flexible and broader. They got better accrual, and therefore had the opportunity for this iterative analysis of responders and non-responders. And some of those principles have gone into the combo match, which is the idea of combinations, the idea of needing preclinical evidence. And it's more flexible because it will use commercial labs. And the idea was that there would be cassettes from each of the different clinical trials networks, and that we could enroll on each other's studies. And I had worked with Mari Yoe and Angelina to develop what I thought would be a really good combo match trial. Selumetinib, a MEK inhibitor, plus tovorafenib, a pan-RAF inhibitor, based on Angelina's beautiful data tumor regressions in rhabdomyosarcoma, not a low-grade glioma, using this combination. And we got this all the way through the combo match process, which I was really interested in because of the potential to enroll children and AYA on adult trials, except in the end, the majority of these are actually histology limited to histologies that don't happen in kids, or use agents where there's no compelling data because the target isn't relevant. And in the end, I can't run my study because it seems that there's overlapping toxicity and there's no mechanism for dose finding in this study. So it's moving back to COG into developmental therapeutics. So there's new legislation that hit committee last week called the Proposing Hope Act. And it's going to do two things. One, it's going to require, it's going to potentially give the FDA leniency to require that industry now do combination studies. And by the way, it will also reauthorize this voucher program that's about to sunset. But in the end, I bring it back to this. I was a fellow before the most important thing that probably has happened in solid tumors since I have been a fellow is the development of anti-GD2 antibodies for neuroblastoma. For solid tumors, this has been the most impactful change that I have seen in the treatment of childhood cancer. And this was drug development by pediatric oncologists for a pediatric disease. There were 2 drugs developed in this way. And this is what you're all here for. So these, again, my top 10 strategies: legislation, mandates, vouchers, the RACE program, target prioritization. It's got to be the most relevant for kids. We don't have enough kids. Biomarker enrichment, but we need to further refine with our trials. Inclusion of kids, collaboration with industry, formulation, prioritization, novel and efficient trials, combinations, and pediatric-specific drug development. And so I think I tried to acknowledge everybody as I went through this, and just wanted to acknowledge the NCI for the match and the combo match. And sorry I went over.
Yeah, you didn't go over. There's still 15 minutes until lunch is served.
Really?
So, so there's actually time for really a very nice discussion, which I hope we'll have now. First of all, thank you for really an amazing talk. And I think that your expertise that you've garnered over the years allowed really very, very important aspects to step forward, or you highlighted aspects that are significant challenges. One of the things that you addressed had to do with the fact that you looked at 200 drugs on the ODAC committee in terms of where they're going to be potentially helpful for pediatric cancer. Another way of looking at that sort of similar issue is a number of years ago, a collaborative project between St. Jude and WashU did sequencing of hundreds of pediatric cancers and came up with a list of what they thought were literally hundreds of targets. And Jim Downing, who's the chief executive officer at St. Jude, was asked, so what does this mean? We've got all these new targets that you think are going to be valid in all these different cancers where, you know, they might occur in a handful of patients each year. I mean, it's a huge problem that we have. I'm going to leave the commercial part of it out of it, just come back to the drug development part for a second. You might have a dozen, 24 patients with these very, significant drivers of cancer. So they asked Dr. Downing, does this mean one postdoc, one mutation? He said, absolutely. So just think about that for your growing careers, and think about that for what it means for research and research support, is that we've got huge amounts of sequencing data right now in the internet. It's all there. We don't need to sequence a whole lot more to know what these targets are. Your comments on how do we begin to think about prioritizing some of that?
It's a tremendously difficult question that you pose, and I I want to get my hands on it, but I have been told that Gilles Vassal, who runs Accelerate, has developed a schema of the number of patients we have helped with these targeted therapy approvals versus the overall childhood tumor burden of disease and how those that we haven't helped. And these targeted therapies, they help these very small fractions of patients. But I would say every fraction counts. But at the same time, you want to hit the most common. And clearly the MAP kinase pathway is important, and we need to figure out how to hit it harder. And in fact, I am really excited about the pan-RAS inhibitor. RAS sits above RAF and is the first oncogene ever to be described and was before now considered untargetable. But with new molecular glues and PROTACs and things like that, these untargetable proteins that we've known about forever, P53 being another obvious one, may actually be targetable. I would go personally for the ones that underlie the most cancers first. That's one route, and that would be the one that attracts industry, right? But for developing a career, I agree with you. Pick small populations out. Certainly these immunotherapies, these CAR Ts, we can't mass produce CAR Ts yet. We may be able to over time, but certainly the small trials, the important targets need to be developed now before we have that technology.
Great questions, lots of questions. Okay, so let's start over here with Dr. Okay, Christine, yeah. Yeah.
So thank you. That was an amazing sort of overview and talk, and depressing and amazing. So one thing I noticed when you have your list up of the 30 most recent approvals is that every single one of those drugs works in an adult indication. And obviously that's why industry pursues it, and then when their hand is forced, they develop a pediatric plan.
Except the last two, right?
Yeah.
Naxitamab and DFMO.
True. So my question is, what can we do as a field? Like, what does the pediatric oncology community do to rescue these orphaned drugs that have had tons of money and development put into them for adult cancers where they failed, but there's data suggesting that they may work in pediatric indications?
I'm so glad you asked that question because I mentioned it to Yuli before too. That is the story of tovorafenib, right? Tovorafenib, this pan-RAF inhibitor, was shelved by Takeda. And Sam Blackman, a pediatric oncologist, created a company called Day One with this precise goal: to rescue assets that have been shelved for kids. And then there is an orphan drug development pathway. It's just that Big Pharma isn't interested in going that route. And he brought it all the way there. And I hope we see more of that model. The other model is the NCI. And the NCI, as you know, was really the sponsor and the developer of dinutuximab until it was FDA and close enough to FDA approval that it appealed to United Therapeutics to buy and develop. And so there are pathways, and they're becoming more common, and hopefully the success of Day One and tovorafenib will attract other people, small biotechs, to start this process of rescuing drugs, focusing on a small but important population, getting the orphan drug designation indication, and then when the drug is on the open market, people will discover what else it's good for.
Thank you again for a fascinating talk. My question was like the great example you gave of the approach of a trial that was iterative and could learn a lot in the same way, but being more, I guess, broad and asking a lot of questions. So how can we better design trials, I guess, to incorporate something like that as a, with all the effort it takes to get to a trial, can the trials be made to be more flexible in that sense that you can learn iteratively in the span of a study, perhaps, be able to enroll, the patients that we need to enroll? Again, I was wondering if that would help with a lot of the challenges of, when we're dealing with rare diseases, fewer patients. How can we best, learn the most we can from one trial?
I'm so glad you asked that, Vandana. I brought that up at ASCO over the weekend. I think we need to get CTEP to recognize that the old trial designs are not going to work in the new era, and that we need for statistical methods to change in the same way that we need new drugs, right? We have new drugs, they need new methods, that we can interpret data and at least have a ongoing hypothesis that over time might prove to be wrong, but gets the drugs to kids and answers the questions. And I'll just give you the example. I raised this over at the meeting at ASCO when we were discussing neuroblastoma and whether we can really compare protracted immunotherapy versus tandem transplant. And I said, how are you going to randomize? Who would agree to that kind of a randomization? Nobody. It's not feasible anymore. The patients won't do it. And so we need a synthetic control of all of that data that we've collected for the last 10 years that immunotherapy's been available, but they've been giving the tandem transplants, and use that as the control arm, and have a single arm study and compare it to those historical controls that you will continue to build with the current study. And I was told, when I asked the question, do we really need a randomization step, that they went to CTEP and CTEP said no. So I think it's going to be patient advocacy and clinical trials and academia advocacy that say it's not feasible anymore. We need to change the way we do things.
I would add to that just a little bit, because this was our experience. With some of the phase 1 studies, it used to be traditional that you had 3 patients on a dose level, and you had to start at a dose level so low below whatever toxicity you might have seen in your preclinical models. And now they're starting to allow one dose level per, one patient per dose level until you get to a point where you start to get some sort of signal. And that makes a huge amount of sense, because now, instead of exposing drugs at incredibly low levels to 9 to 12 patients, you're exposing 3 or 4 patients, and that's letting you then give higher doses to those other patients that could potentially benefit. So there's a lot of things in that space that I think are important. Brigade, you had a question. Oh, you lost the microphone. Oh, let's go over here. Okay.
Dr. Glade, I thought this was an amazing talk. Thank you. I had a question about the priority review voucher program. Since you mentioned that dinotuximab was one of the most significant breakthroughs in solid tumors, United Therapeutics took advantage of that program. So could you comment on why this was discontinued and whether there is a new initiative on the horizon? Is it all money?
So it's not discontinued. Okay, it's sunsetting in September right now, September of this year. And what it means is that you can't sort of apply for that rare disease designation after September 30th of this year unless the Give Kids Hope a Chance Act is passed, in which case it will be reauthorized and there will be no gap. So it hasn't yet expired. But it will expire if Give Kids Hope a Chance doesn't pass. So reach out to your congressman.
I echo, it was a great talk and really brings back many memories, because for me too, like on crizotinib, I saw my first response to a phase 1 agent within a week, and that was just like a miracle. So lots has happened. But I was wondering, for combinatorial strategies, do you think people will down the road combine tumor microenvironment and immune-targeted therapies in like the early setting to see, like right now we typically don't do these combinations, and is that something that would be done through institutions or CTAP or?
Well, I'm hoping that what we could try to pitch as a reasonable design would be, if you need to do dose finding or if you need single agent data just to finish your package, right? It's short. It's a week, 1 week, 2 week. Then you can add on, and you can do intra-patient dose escalation if it's too low a dose. You can do intra-patient, add the next agent on. And you know, these trials, I haven't seen them, but these are the kinds of trials that I think need to be done. But we need our statisticians to help us figure out how we're going to be able to use that data in order to get it to the finish line.
Hi, that was a fabulous talk. I think you said you gave it for the first time, so you should please keep giving it. The historical review of all the legislation, I think, is particularly important for folks who don't have that awareness. And I had the opportunity to be part of a PEDS ODAC meeting specifically on the FDARA. And I think you know, you presented really nice data that the FDA is even trying to get a handle on about whether these initiatives have actually made a difference. So I have two just quick questions and one sort of clarifying comment. I think the molecularly targeted can be sometimes a little bit misleading, and I wanted to clarify and confirm that that also includes CD19 and immune-targeted therapies, just because the nomenclature is molecular, but if you're in immunotherapy, you sort of forget that that includes other therapies as well.
That you're 100% correct. I should really just call it targeted therapies, and the idea is really that you should have a biomarker of the patients who are most likely to respond, and that would cover targeted therapies. Molecular, sometimes, as you're right, you know, you might want to say targeted immunotherapies. Targeted molecular therapies are generally pathways, but this was a list of everything. Immunotherapies and molecular drugs.
And I just wanted to make that point because I think as it relates to even the concept of either targeting very rare diseases or something as ubiquitous as CD19, this list is extensive and they've done a fabulous job, and I just wanted to make sure everybody had that awareness. My second question for you is that, you know, when I hear talks like this, and I think we've all had the experience, we all get really fired up. But it is a little bit like preaching to the choir because there's only so much that we can do with academia. I think we've struggled and sort of maybe butted head with pharma. Maybe the pediatric, you know, it's not only the drug development, but the lack of profit. What can we do in terms of engaging with our patient advocates and other folks who might be able to really help make a difference to advance the field? Because I do feel very much that we're stuck.
So I think you are correct that we have underutilized our patient advocates, and I think the legislation has worked because patients show up at the Hill and their congressmen listen, right? And so I also think that I, in particular, but all of us in general, have not been good enough about having the patient voice at the table when you are designing the trial, because there are too many trials that fail. And why do they fail? Because the patients don't want to go on them. And we need to know that beforehand because it's so much effort that we need to address those hurdles before. And we need to engage industry. They should be paying for these trials. They should be helping to cover the costs of the travel and the research-related studies that we need to do. They need to help the patients. They need, you know, we need to be able to make sure that it's not just patients with families with means that make it to clinical trials. We need to improve access to clinical trials. And I think that the patient voice is probably the most important voice, and we have underutilized it. But I also think that we need to, as academicians, sit down with pharma and explain this to them, because I was delighted when I met with a company this past weekend, and they described the trial that they wanted to do, and I said, yes, that's the right trial. We're going to do dose finding. We're going to start with the adult recommended phase 2 dose, and we're going to do it in these patients, and then we're going to do a small exploratory cohort of 2 different diseases, and if we see signal, then we're going to go right to a randomized study. And I'm like, yes. I don't see these in the United States. They do them in Europe, where you go all the way from phase 1 to phase 3 in a single trial. And even in phase 1, they said, oh, and we're going to allow intra-patient dose escalation in phase 1. And I was like, yes. So I do think people are hearing, but we need to be at the same table and not be working in silos.
Yeah.
Sorry.
Thank you. Just an excellent overview. So I've been in this career now for a few years, right? Certainly not like 10. I still don't know who they is, right? Meaning I sit in the room with disease experts. They have this fantastic trial idea, and it goes before them, and they say, no, you can't do that, right? Or like you said, right? You sat down with fantastic data, fantastic scientists came up with an excellent trial, and there's them who said it's a no-go. It's almost, it feels like insurance just saying, no, you can't do that, and then you have to talk to someone. So, forgive me, but who is CTEP, right? Who are, like...
I was going to say, they're probably your colleagues.
Well, I guess so maybe just some education for me as well, right? Like, anyways, I'm just curious in that it's, I hear this kind of refrain, over and over, like, a lot of people feel we should do this, but there's some wizard behind the curtain who nixes it.
Yeah,
and
no,
you don't know,
I totally agree with you, and that's where I think my recommendation was we need to bring patients, because I think them is sort of stuck, just like COG was stuck in this one sort of model, but I think is coming unloose, right? Because with new drugs, diminishing numbers of patients, rare diseases, we need to come up with new methods. We can't rely on the old methods. And we can't just keep building more and more and more on our backbones. We need transformative changes. And I think the voice that will be listened to is actually the patient voice. And so I think we need to bring patient advocates with us when we are on the CTEP calls. Right now, the CTEP patient advocates are actually there to say, they're there to tell you that they wouldn't do the trial, but they're not there to tell you the trial that they want. And I think we need to change the role of a patient advocate. And I think we need to bring industry as well. I think we just need to start being much more multi-stakeholder in our approach and not have this sort of unilateral decision-making that goes on both at, it's amazing. CTEP will say yes, FDA will say no, FDA says yes, CTEP says no. We need to change the process. And we need the methods to also evolve.
There's many layers.
Yeah.
Okay. That sounds good. Thank you very much. That was great. Yeah, it was really great.
Okay. Not exactly what you were thinking, but.
It didn't matter.
Okay.
Yeah, no, it's great. All right, well, thank you again. This really was a wonderful way to end the morning.